Thank you very much, Hilton, for that generous introduction, and thanks to all of you for coming out on this lovely, lovely evening. Astrobiology, to me, isn't a field, it's a calling. As Hilton said, when the press conference was held at NASA headquarters in 1996, it was August 7th, 1996, I remember the day, and there were people on a stage very much like this one, in the auditorium at NASA headquarters saying, we think we have found evidence of life in a meteorite that came from Mars. I thought, wow, this is something I really want to be a part of. And at that time, I had somehow managed never to have taken a biology class. I had managed to avoid it in high school, I had managed to avoid it in college, and by the time I got into a graduate program to get my PhD in chemistry, of course, I didn't care whether or not I knew any biology. So as Hilton says, I started off to learn biology, and it wasn't really self-taught. I found some wonderful teachers to whom I will be forever grateful, and some wonderful courses and programs. And I have been a student of biology ever since, and I think I will be a student of biology to my dying day. So it's really exciting to share this amalgam, which astrobiology is. It's an amalgam of the biological sciences, the astronomical sciences, the geological sciences, combined with chemistry, with physics, and frequently with philosophy and ethics and theology as well, as you're about to see. So thank you all for coming out for this. I hear you ask, what is astrobiology? This is the way I like to characterize astrobiology. It's the study of the potential of the universe to harbor life beyond Earth. Now, that is a question, the potential of the universe to harbor life beyond Earth, that people have asked about and wondered about probably pretty much for as long as there have been people. So here's a quote. This is a quote from Epicurus. He was a Greek who lived around 300 BC. And what he wrote is that there are infinite worlds both like and unlike this world of ours. We must believe that in all worlds there are living creatures and plants and other things we see in this world. A strikingly modern view, modern perspective on the universe. There were other perspectives. This is a perspective of theologian, St. Thomas Aquinas, who lived in the 13th century. He wrote that when it is said that many worlds are better than one, this sort of better does not belong to the intention of God, because for the same reason it could be said that if he made two, it would be better that there were three, and thus ad infinitum. Thomas was not a fan of there being virtually an infinite number of worlds. But as I'm sure most members of this audience know, the number of worlds out there is very, very, very large, and although it is not infinite, it is an extraordinarily large number. Today, we are privileged to be able to bring the tools of modern science to bear on these age-old questions. The three fundamental scientific questions of astrobiology are: how does life begin and evolve? Does life exist elsewhere in the universe? And what is the future of life on Earth and beyond? In order to address those questions, we bring together 5 interconnected areas of science. And this slide is basically an outline of the rest of the talk this evening that you'll hear in the next half hour or so. We're going to start off speaking about the diversity of life on modern Earth. Then we're going to talk about the 4 billion year coevolution of life and our planet. The Earth is 4.5 billion years old, and life has existed on Earth almost since its very beginning. So we're going to talk about that coevolution of life in the planet. We'll then go outward and talk about the diversity of solar system environments. And let me just say that the reason we start with understanding life on Earth is that in order to understand the potential of the universe to harbor life, we really have to understand the one planet which we know for sure harbors life, and that planet, of course, is Earth. And we have to understand the nature of life and at least have some theories about how life came to be on the Earth. And that then informs our study of potentially habitable planets around other stars. So we start by studying the Earth and its life. Then we move out into the solar system to look at the diversity of solar system environments, to understand if, based on our understanding of life on Earth, whether there are habitable environments elsewhere in the solar system to which we can actually send spacecraft. Then, of course, we now know that there are planets around most stars, and so we also inquire about the planetary environments around other stars. And then, of course, one of the most fundamental questions we can ask: how does a planet go from non-living to living, the origin of life? Where and how are the raw ingredients for life made, and how may that transformation from non-living to living? Let's start with the diversity of life on Earth. If you studied biology a few decades ago and you were studying the diversity of life on Earth, you were probably presented some version of this diagram. This is the tree of life in the five kingdom perspective. So each of these sections of the diagram is a kingdom of life. And you were taught that at the base of the tree of life is a form of life called monera. These are simple single-celled organisms that don't have cell nuclei, that don't have compartments in their cells. Then you were taught that the next most complex form of life was also single-celled life, but a form called protists that are larger cells, typically 25 times larger than the monera. And these are cells that have cell nuclei and that have other compartments in the cells. And then you were told that the real diversity of life, however, was in the so-called crown kingdoms of life: fungi, animals, and plants. Now, this perspective on life was developed both by looking at the organisms, looking at the microscopic organisms, these unicellular organisms under the microscope, and then looking at fungi, animals, and plants frequently with the naked eye. And of course, one can see there is a tremendous amount of diversity. And so when one formulates a perspective on the diversity of life based on what you can see, as well as how organisms reproduce, you're led to this conclusion that most of the diversity of life is up here at the top of this tree of life among the fungi, animals, and plants. Well, this is a modern version of the tree of life. This version is based not on how organisms look or how they reproduce, but this is based on their genetic information. That is, this is based on their genomes. This is based on one particular gene that is present in all living organisms. And it turns out that all of the fungi, animals, and plants are all represented by those three twigs on this genetic tree of life. We are here. Move that E up to there. That's us. That little twig, which is labeled Homo, represents all the animals. This little twig right there, which is labeled Zea, represents all the plants. And this one here, labeled Coprinus, represents all the fungi. This domain of life, this tree of life, shows that there are in fact three domains. This domain is called eukarya. All the rest of the eukarya here, those are the protists. It turns out that the monera are everything up here. The monera are actually two distinct domains of life, one that we call bacteria and one that we call archaea. So the monera turn out to have the bulk of the genetic diversity of life. What a surprise. Instead of being uninteresting and all the same, it turns out that the bulk of the metabolic diversity of life is here in the monera, in bacteria and in archaea. And it turns out as well that this genetic diversity carries with it a tremendous amount of physiological diversity and metabolic diversity. And it is that physiological diversity and metabolic diversity that we find in the bacteria and the archaea in particular that allows life to inhabit virtually every habitable niche on Earth. So places that we never thought were habitable are in fact inhabited. So, for example, we thought that the floor of the ocean would be uninhabited. There would not be ecosystems on the floor of the ocean because, after all, everyone knows that that life requires light, and it's dark on the floor of the ocean, so there's not going to be life down there unless something swims down from above, in which case it's going to swim back up. Well, in fact, that's not the case. I think probably most people in this audience know that there are thriving ecosystems at hydrothermal vents on the seafloor. This is a picture from one of them. These are tube worms. These are typically 1 meter or 2 meters long, and if this was a larger picture, you would be able to see that there are little crabs and shrimp that feed off of the tube worms, and this ecosystem is powered by energy that is coming out of the earth combined with oxygen that is in seawater that's brought down from the surface, and we're going to talk more about oxygen in just a little bit. This is a picture of Grand Prismatic Spring in Yellowstone National Park. There are many such springs. They are very hot. Typically, the temperature in the center of the spring might be around 90 degrees centigrade, which is very close to the boiling point of water. Some of these springs are acidic, approaching the acidity of battery acid. So you have boiling sulfuric acid. And the water in the center of these is crystal clear blue. It surely looks like it must be sterile. And guess what? It's not. It's teeming with microbial life. And again, it is this genetic diversity that allows these environments to be inhabited. This is a rock from the wall of a dry valley in Antarctica where it never rains, where there doesn't appear to be any accessible water, and these are living organisms, microbes, right underneath the surface of that rock who manage to extract water just from the atmosphere in these very, very dry environments. So wherever life can exist on Earth, it does, and it is because of this tremendous amount of genetic diversity that we see. I mentioned that the genetic diversity corresponds to a great deal of metabolic diversity, and what we have in this diagram is an illustration of how many metabolisms there can be for life. So let's just think about our metabolism for a second. If you don't photosynthesize, does anybody in the audience photosynthesize? Okay. No one photosynthesizes here. So what that means is that we need to eat a fuel, and we call those fuels reductants, and we need to combine our fuel with something that we call an oxidant. Now, us humans and all animals use as our oxidant oxygen, which has the chemical formula O2, and we see that right over there. And our fuel is glucose. We eat sugars, and we convert starches into sugars, and we combine that sugar with oxygen, and we breathe out carbon dioxide and water. That's our metabolism. Now, the amount of energy that an organism derives from its metabolism is shown by the vertical distance between the fuel or the food on the left and the oxidant on the right. So you notice glucose is way up here, oxygen is way down here. The vertical distance here is almost as large a vertical distance as you can have on this diagram. And what that tells you is that we have a very energetic metabolism. It produces a lot of energy. It's what enables us to grow big and strong. There are lots of organisms that use metabolisms that are not quite so energetic. So for example, one metabolism that is very common is, and metabolism that particularly may have been very common on early Earth, is a metabolism where hydrogen is the fuel and carbon dioxide is the oxidant. And there are organisms that use that combination, and they produce methane. They're called methanogens. And there are lots and lots of methanogens in this room with us. Anybody know where? In our guts, exactly. So again, this just illustrates how many environmental niches there are for organisms with different kinds of metabolisms. And in fact, if you take any fuel on the left-hand side of this diagram and any oxidant on the right-hand side of this diagram, if the arrow connecting them goes down to the right, then there is an organism someplace on this planet that uses that metabolism. So that shows you the range of metabolisms that are available to organisms, and that is really central to enabling organisms, particularly microbes, because we are largely talking about microbial metabolisms here. This is what enables so many of these environmental niches, including our guts as an example, to be inhabited by microorganisms. So let me move on now to the coevolution of life and the planet. What you're looking at here are pictures of rocks that are 3.5 billion years old. Now, the Earth is 4.5 billion years old. These are among the oldest rocks on the planet. These are rocks in Western Australia in a region called the Pilbara. And take a look at these structures in the rocks. There is a pocket knife for scale. This structure is a couple of feet high, maybe 2.5, 3 feet high. There's two of them. They grade gradually into this larger dome. Here you see another structure, not quite the same shape, rather more conical. These are fossilized microbial communities. These were photosynthesizing communities of microbes that grew upward toward the sunlight. This is why they have this domed shape and this conical shape, because they were trying to grow upward to get the light. These have been studied in great, great detail, and we're quite certain that they are, in fact, evidence of rather thriving microbial communities 3.5 billion years ago. Now, one of the things that helps us appreciate that these are, in fact, evidence of biology is that we can see the same kinds of communities alive today. This is also a picture from Western Australia. This is from Shark Bay. This is just a few hundred kilometers away from the Pilbara where this was taken. Just look at the similarity here. This is 2.5, 3 feet high. That's also about 2.5, 3 feet high, just about the same size. And in fact, we believe that this is a modern version of that. Life has existed for a long time, at least 3.5 billion years. We think life may be older than 4 billion years. One of the early forms of life, but by no means the earliest, are these organisms. This is, of course, a picture of modern cyanobacteria taken in a laboratory. But these organisms were around on Earth at least 2.5 to 3 billion years ago, possibly even longer, possibly as much as 3.5 billion years ago. And these are tremendously important organisms. They photosynthesized, but the most important thing is that they were the first oxygenic photosynthesizers. These were the very first oxygen producers on our planet. Now, the production of oxygen has been a profoundly important aspect of the history of Earth. All of the oxygen in Earth's atmosphere is the result of life. And oxygen wasn't around on Earth until these organisms evolved. These are rather complicated photosynthesizers. There were simpler photosynthesizers that didn't produce oxygen hundreds of millions of years before cyanobacteria came along. So, one of the things that we can look at is we can look at the history of oxygen on Earth, and this is a very simplistic perspective on that. This side of the graph, P stands for pressure, and so this is the pressure of oxygen in units of the current atmospheric pressure on Earth. So atmospheric pressure on Earth is 1 atmosphere by definition, and 20% of our atmosphere is oxygen. So this is the modern level of oxygen on Earth. And this is a scale in which each unit goes down by a factor of 10. So prior to the development of cyanobacteria, there was 0 oxygen. It was all the way down. And then at some point, and in this particular graph, it suggested that it was maybe 3.2 billion years ago, cyanobacteria evolved. They started generating oxygen. But oxygen didn't immediately start filling up the atmosphere. There was a lot of reduced material. Remember what was on the left-hand side of that metabolism diagram that we called reductants? There was a lot of that kind of stuff around, and that just eats up oxygen chemically. So there was a lot of reduced iron in the oceans, for example. And that reduced iron just soaked up this oxygen. And in fact, it made, in the process, the iron ores that we utilize today. In fact, in a way, you can say that cyanobacteria were responsible for the industrial age because they provided the oxygen that led to the deposition of the ores that we mine today as the primary sources of iron. After all of those reductants got soaked up, or soaked up the oxygen, then finally oxygen rose again, something on the order of 2 to 2.5 billion years ago. We think it was about 2.4 billion years ago. And it rose up perhaps to modern levels, perhaps it dipped down again. But the point is that the oxygen that we see today on Earth was not always there. Now, this is really important when we talk about exoplanets, because you will frequently hear people, when they're speaking about exoplanets, say, well, if we can find oxygen on an exoplanet, that means that there must be life, because we know that all the oxygen in Earth's atmosphere was produced by life, and therefore, if we see oxygen on an exoplanet's atmosphere, well, there's a good chance that it was produced by life. Well, that's an okay argument, except that if you had observed Earth, if you were an astronomer on that exoplanet looking back at Earth 3 billion years ago, or 3.5 billion years ago, you wouldn't have seen any oxygen. How could you have told that there was life on Earth? So when we look at exoplanets now, we have to think very carefully about what kinds of characteristics would tell us that there was life on that exoplanet. Because Earth has had many different characteristics over the course of its history. The abundance of oxygen in its atmosphere is just one of those. And there are many others, the diversity of solar system environments. And I'm going to talk about 4 objects in particular. I'm going to talk about Mars. I'm going to talk about Europa, a moon of Jupiter. And I'm going to talk about 2 moons of Saturn, Titan and Enceladus. So here's a picture of Mars. This is one of many pictures. It was sort of a random picture. I chose it because it's just visually so striking. But what it shows you is it shows you the remarkable complexity of the geology of Mars. You see many, many layers. You see a tremendous amount of erosion, a very, very complicated geological history. One of the things we've known about Mars for many years, actually since the first missions to Mars in the late 1960s and the early 1970s, is that there has been a great deal of water flowing on Mars, at least in the distant past. Here is an example. This one happens to be taken from a spacecraft that ESA flew to Mars called Mars Express. ESA is the European Space Agency. And this one shows channels. These are channels that were almost surely carved by flowing water, flowing downhill into this lower area. You can see an area here where the support in the ground seems to have been removed. And we think what happened here is there was subsurface ice, melting occurred, the ground collapsed, the ice melted, and water flowed out in a channel into this lower region. And you can see evidence of many other channels. And this is something we've known about Mars for many years. Starting in the 2000s, we had a series of landers on Mars. This is just one of those, the Opportunity rover that landed on Mars in 2004. This is the Opportunity rover just having rolled off the landing system that delivered it to Mars. This landing system used airbags. You can actually see the airbags if you look carefully. They are retracted and folded under. And you can actually see the bounce marks because this was surrounded by airbags when it landed. And those airbags deflated after landing. You can actually see the bounce marks there where the spacecraft bounced before depositing the Opportunity rover on the surface of Mars. And we sent the Opportunity rover to a landing site that we saw from orbit showed evidence that there were minerals that were formed in water. And sure enough, when we looked at the landing site, what we found are these small blueberry-sized spheres, which we call blueberries, although they're made out of rock. They're made out of an iron ore called hematite. And these are, in fact, examples of the kind of rock that gets formed in water. And these blueberries are embedded in this rock. And as this rock erodes, these blueberries basically fall out of the rock. And this happens to be a little depression, which the team that was running the rover called the Blueberry Bowl. Now, we sent other landers to Mars. This is the Phoenix lander. An image taken by a camera on an arm of the Phoenix lander looking under the lander. Now, unlike the Opportunity rover, the Phoenix lander was sent to high latitudes close to the North Pole of Mars, and it used retro rockets rather than airbags as its landing mechanism. So this was more of the classical lander that you expect, that fires a retro rocket and then lands on legs. But when you fire a retro rocket, one of the things that you do is you blow away some of the surface immediately underneath the spacecraft where you're landing. So the retro rockets from the Phoenix lander blew away a few inches of Martian dust, and look what it exposed: ice right underneath the surface. This is just one of many pieces of evidence that there is lots and lots of water on Mars today, most of it frozen, but not all of it frozen. And here's an example of water that's not frozen and was there on Mars and is probably still there on Mars. These are the struts, the legs of the Phoenix lander, and look at these droplets. The way those droplets formed, we believe, is that there is a salt in the surface of Mars. It happens to be a salt which bears a chemical resemblance to common table salt, sodium chloride. This is not a chloride, but it's a perchlorate. It's got chlorine in it, but the chlorine's combined with oxygen. And that salt, like table salt, absorbs water very, very readily. But perchlorate really absorbs water. So even though there is very little water in the Martian atmosphere, there is ice right underneath the lander, there is some water vapor in the atmosphere, even though it's very cold. And that salt absorbed enough water vapor out of the atmosphere to form liquid water droplets on the struts of the lander. So not all the water on Mars is frozen, and there's other evidence that there may be at least small amounts of liquid water relatively close to the surface in certain areas that get heated by the sun. The most recent lander that we've landed on Mars is the Curiosity rover that landed on Mars in 2012. It's taken a number of selfies. We sent it with a selfie stick. It's got a long arm with a camera on the end of it. So you don't see the selfie stick here. The selfie stick, seriously, of course, is an arm that carries several scientific instruments, and it enables the rover to place these scientific instruments up against rocks to do very detailed analyses. But it can also take a picture of the rover itself, which it has done here. And one of the very first things that the Curiosity mission discovered at its landing site, which we thought was a place where there had been water, at least in Mars' distant past, a place called Gale Crater, that's spelled G-A-L-E. And sure enough, one of the very first things that the Curiosity rover found were stream deposits. And the way that we know this is a stream deposit is that it's a conglomerate of rocks. This isn't a solid rock. This isn't a piece of bedrock. This is more like a piece of cement where you have lots and lots of little pebbles that have all gradually gotten cemented together just by the natural processes of being exposed to weather. And in fact, this and other information that has been gathered from the Curiosity rover tells us that it probably landed in an ancient lake bed toward the edge of Gale Crater. And it was a lake bed that was fed by some streams that were coming off a central mountain that I'll show you in just a second, called Mount Sharp, and coming off the walls of the crater, flowing down into the crater at a time when there was much more liquid water on Mars than there is today. Curiosity was sent to Gale Crater because in the center of Gale Crater is this mountain called Mount Sharp. Mount Sharp is roughly the same size as Mauna Kea, measured from sea level, not from the floor of the ocean. This is a large mountain. And Curiosity is in the process of climbing up into the lower reaches of this mountain. It's going up toward this side and moving up in that direction. And as it goes up, it is studying the rocks to understand what was the history of water in this part of Mars, because the lower regions of the mountain are, of course, the older regions, and as you go up, you go to successively younger regions. And so the mountain serves as something of a time machine, and as Curiosity climbs up the mountain and studies rocks at different levels on the mountain, we will be learning about different periods in Mars history. This is a picture I just like to include just because I just find this picture so powerful. It makes me feel that I'm standing there on Mars. That is the wall of Gale Crater. This is a sand dune. We're looking at the downwind face of a sand dune. This is the kind of terrain that the Curiosity rover is traveling over. We try very hard not to drive the Curiosity rover into a sand dune because that would not be good. But in fact, we can get it up really close, as you can see. So, and this is just part of a 360-degree panorama. You can find this, the image from which I took this, and many, many other images from Curiosity and the other missions on the web. They're not too hard to find. Typing Mars images into Google will be a good start, or just typing in Curiosity rover mission. Further out in the solar system, to Europa, a moon of Jupiter. If you looked at Europa with your naked eye, it would look something like this. It would almost look like a cue ball in a billiard set. If you enhance the resolution, if you enhance the contrast a little bit, you would find that it looked a little bit more like this, and you would see that there were mottled areas. You would also see that there were some linear areas. This is an object roughly the size of Earth's moon, so this is not a small object. And if you look at the surface more closely, in fact, it was my PhD thesis back in 1972 that told us that all of this white material is frozen water. That's water ice. And when we look at the surface more closely, and this was from the Galileo spacecraft in the late 1990s, we find that the surface, particularly those mottled areas, are areas in which the icy surface appears to have been heated and broken up. And you can see that chunks of ice have sort of drifted off. And because one of the things that is very common on the surface are these ridges, which are frequently double ridges, you can actually sort of put the pieces back together. So for example, that double ridge was once connected to that double ridge, but it separated and this piece of ice floated off. If you're having any trouble seeing the topography here, this face is facing the sun, and there's the shadow. So the sun is coming in from the right, and there is the shadow of this particular piece of ice sticking up. This is sticking up perhaps 100 meters, and this is perhaps a kilometer or 2 kilometers across. What we think is going on here is that there is a relatively thin frozen ice covering, and there is a liquid water ocean under that ice covering 100 kilometers, 60 miles deep. There is as much water in this global ocean on Europa, we believe, as there is in all of Earth's oceans combined. What a fascinating place. Wouldn't you like to get into that ocean and find out if there's anything swimming? I sure would. Let me move on to Saturn. This is Titan, a moon of Saturn. It's covered by a photochemical smog, sort of what I used to see when I was observing at Mount Wilson, which looks down on Los Angeles in the 1970s. And I looked down during the day, and what I saw was something that looked very much like Titan. And not until the sunset did I start seeing the streets, because the photochemical smog at that time was very great. But we can see through haze of Titan with radar, and we've done this with the Cassini spacecraft. And what we have found is that there are lakes and rivers on Titan. This is not a radar image. This is actually an image taken directly by the Huygens probe, which was carried to Titan by the NASA Cassini spacecraft. The Huygens probe was built by the European Space Agency. And this is an image showing river channels on Titan with the evidence of rain. And in fact, we know that there are storms on Titan, and those storms involve rain with rivers that then flow down and fill up lakes and small seas. However, the liquid is not water. It's way, way too cold for water. Does anybody know what that liquid is? Liquid methane, exactly. We have methane seas, methane lakes, methane rivers, and methane rain on Titan. Now, one of the questions I sometimes get asked after a talk like this is, well, we're always talking about water and looking for water as signs of life. Could life exist in some other solvent? Well, the answer is yes, life could exist in other solvents, and one can have some interesting speculation about whether or not there could be life in methane as a solvent. And one mission proposal is to land on one of the methane lakes on Titan and investigate what's in that lake and see, is there perhaps evidence of life in methane lakes on Titan? And this is a picture that was not supposed to ever be taken. This is a picture from the Huygens lander after it landed on the surface of Titan. It wasn't supposed to work after it landed on the surface. It was being lowered on a parachute, but it was designed to function only while it was descending on the parachute. And here we have a picture of rocks in the foreground. These rocks are made of water ice on a surface which in fact is a surface that has probably been wetted by methane, although it's a little bit hard to tell whether it was wet with methane at the time that Huygens touched down on it. Another moon of Saturn, a much smaller moon called Enceladus. And Enceladus is another moon that we have learned has a subsurface ocean and an icy surface. But on Enceladus, the ocean is in part escaping into space. There are jets coming out of the south polar region of Enceladus. We have been able to study these jets from the Cassini mission. We've been able to actually fly through the jets and measure their composition. We know that the jets are mainly composed of water, but there are salts in the jets. There are also organic compounds in the jets. And there is evidence of the kind of interaction of water and rock that we think might have helped life get started on Earth 4 billion years ago. And there is evidence of some of that same chemistry going on Enceladus. And there are some people who think that Enceladus may be one of the most likely candidates in the solar system for a body that might harbor life in an ocean beneath its frozen surface. Let me move on now to planets around other stars. The first planets around other stars were detected in 1995-96, and as Hilton mentioned, were part of my inspiration for going into astrobiology. And the Keck Observatory has been absolutely fundamental in these discoveries, particularly in discovering planets by the Doppler method. In the Doppler method, And in both methods of detecting planets that I will speak about, and I think people in this audience know about, you don't actually see the planet. The planet is unseen. But what you see is the gravitational effect that the planet has on the parent star. As the planet moves around the star actually also moves, and we can see the star moving alternately toward the observer on Earth and away from the observer. We call this a blue shift. We call this a red shift. We can actually detect that. This is a sample spectrum taken with one of the instruments at the Keck Observatory, the high-res instrument. And it is by this means that hundreds of planets around other stars were discovered, and hundreds of them from the Keck Observatory. The other method is the transit method, and I know you've heard about that recently as well. This is the method that one can take advantage of when the orbit of a planet is aligned so that the planet, as it goes around its parent star, passes in between the star and you, the observer. When it does that, as shown in this artist's conception, it blocks out a little bit of the light. This can be done from the ground, but in order to detect Earth-sized planets this way, which block out only 1 part in 10,000 of the light from the planet, so you have to detect a dimming of the star, the light from the star. You have to detect a dimming of the light from the star of 1 part in 10,000. That's really small. You need to go into space to do that. The Kepler mission is doing that, and the Kepler mission has discovered thousands of planet candidates, most of which are real planets, but frequently you need ground-based observations to confirm that something that Kepler identifies as a potential planet is, and much of those ground-based observations are also done on Mauna Kea, and particularly at the Keck Observatory. What we are really interested in, and what Kepler in particular is focused on, is finding planets in the habitable zone of their parent stars. The habitable zone is defined as the distance from a star at which liquid water would be stable on the surface. So it's sort of the Goldilocks zone. If you're too close to your star, it's too hot. If you're too far away from your star, it's too cold. But in this green region, liquid water could potentially exist. And of course, the habitable zone is at a different size depending upon whether you're looking at a really hot star, or a star like the Sun, or a really cool star like the red M dwarfs that I'm sure you have heard about. So Kepler is particularly focused on finding planets in the habitable zones, and the observatories on Mauna Kea, particularly including Keck, have played an important role in following up in those observations. And today we know that someplace between 1/5 and 1/4 of all Sun-like stars have an Earth-sized planet in their habitable zone. And this is an estimate, of course, because we're just beginning to discover these. But what we are learning is that Earth-sized planets, potentially rocky planets, appear to be common. Most stars, or at least something on the order of 20% of Sun-like stars, and perhaps even larger percentages of other types of stars, may have planets that are roughly Earth-size and could potentially be habitable. The last of these topics is the origin of life. I'll say just a few words about this. One could talk about the origin of life for a long time. It is one of the great mysteries of the universe. One possibility that we are aware of is that the raw materials of life may have come from space. When we use telescopes, such as the telescopes on Mauna Kea, particularly the radio telescopes, and look, for example, at interstellar clouds, we find evidence of the molecules that ultimately are important for life. We then also can study meteorites, and that is samples of interstellar material that coalesced into solid bodies around forming stars and eventually fell to the surface of the Earth. This is a meteorite that fell to Earth in 1969 in Australia near a town called Murchison, so it's called the Murchison meteorite. It's a kind of meteorite we call a carbonaceous chondrite because it's got carbonaceous material in it. And it turns out that that carbonaceous material includes amino acids and nucleic acids. Amino acids are what make up our proteins. Nucleic acids are what make up our DNA. So some of the raw ingredients for life are being made in interstellar space and in circumstellar space in planetary systems as they are forming. And that material is being delivered to Earth today. And it was being delivered to Earth in much greater abundance 4 billion years ago, when there was a lot more of this debris left over. So Earth may have been seeded with the raw materials that it needs for life, possibly from space. The raw materials for life can also be made at hydrothermal vents, and we have experiments that we're doing in the laboratory to show how some of the raw ingredients for life can be made, and even how chemical systems can develop at hydrothermal vents that emulate cells, that actually emulate in small compartments in the minerals that form, that actually emulate a primitive cell, even though it is strictly a geochemical phenomenon at a hydrothermal vent. So we think that life could have arisen deep, deep in the ocean at a hydrothermal vent, or perhaps life could have arisen back at the surface, but in a tide pool. One of the problems in the origin of life is that many of the molecules that we need for life are formed as polymers. That is, there are individual smaller molecules that we call monomers, and they have to link up together to form a polymer. Well, when you link up monomers to form a polymer, commonly what happens is a water molecule is extracted from those two monomers in order to join them. Well, it's very hard to extract that water molecule when you're bathed in water, as you are in an ocean. So one of the things that helps is if you can dry out sometime, because as you dry out, the environmental water goes away and it becomes easier to form those polymers. And sometimes the rocks that you are, that the molecules are resting on and attached to are clays that are formed by the action of water on rock. And those clays can be catalysts for the formation of those polymers. And so a tide pool, and this is a tide pool in Portugal today, but tide pools which get wet at high tide and then dry out at low tide as the sun shines on them and then get wet again at the next high tide, they may have played an important role in the origin of life. So we don't know for sure how life began on Earth, but we have a lot of theories. So I'd like to just finish up with the question, how would we recognize alien life if we found it? Now, it would be really nice, of course, to have a tricorder and to have Bones there and Jim. And, you know, Jim could ask Bones, well, Bones, is that a life? Is that a life form? And Bones could say, yes, Jim, that is definitely alive. See this reading here on our tricorder. Well, of course, we don't have a tricorder. Now, there is also the Supreme Court theory for life, and that is we'll know it when we see it. Let me close this talk with another possibility. Let's just think about the types of molecules that life uses. Again, the only life that we really can say things about definitively is life on Earth. So let's just start with life on Earth. And let's just sort of generically think about the type of molecule. Well, we know that life uses several types of molecules. We know life uses sugar. Glucose is an important one. We use some other sugars. We can eat fructose. We can eat sucrose. So there are a few other sugars. We have a particular sugar in our DNA called ribose, which is a different kind of sugar, but it's important for us. We use amino acids in our proteins. We use nucleic acids in our genetic material. But those are very specific types of molecules, and we use very specific versions of those molecules. So we can think of a biological distribution of molecules as being spiky. We would have, you know, a few different kinds of sugars, a few different kinds of nucleic acids, a few different kinds of amino acids. So it would be a spiky distribution. So if in some sense we plotted the abundance of different types of molecules, and this is just the relative number of those molecules of the relative abundance, we would find a spiky distribution. Abiotic processes, in comparison, tend to produce relatively smooth distributions. That is, they'll produce all kinds of amino acids, all kinds of sugars, all kinds of nucleic acids, not just the kinds that we use. But let us say that we went to Mars or any other planet where we could access material, and we looked for molecules without any presuppositions, and we found a spiky distribution of molecules, but it was a different spiky distribution than the spiky distribution for Earth life. We might think that that's a clue that there might be biology at work, and we should look deeper into that question. And with that, I would be happy to answer any of your questions.